There are two lecture note sheets that I have provided for you, page 1 and page 2. By now, you know that you could choose to either print it out or write it in your own notes in the sequential format using subheadings so that when I take a look at your work, I can see what you have done. Now, I do want to make a point, is that for this particular lecture, there is an exit challenge. The exit challenge is not obvious right now because it is not printed on both of these sheets. It is a separate assignment that you will see towards the end of the lecture. So what were some of the key experiments that led to the interest in identifying the hereditary material? Up until this point, you have learned about Gregor Mendel's work. You have also learned about Sutton Boveri's work. And you have also learned a lot more about Morgan's experiment that led to the discovery of sex-linked traits, which ultimately led us to complete a better understanding that there is such a factor or unit that is being transmitted from one generation, that is being passed from one generation to the next. But up to this point in history, there really is no indication as to what this molecule looks like. And that is where the focus of this lecture is about. So let's take a look at this. This is an image of a patient who is suffering from a respiratory disease. This particular individual is suffering from Streptococcus pneumoniae. It is pneumonia that's caused by a type of bacteria that can come in two different forms, a smooth strain or a rough strain. Now, these two generic terms that are used to describe these two strains of bacteria, the smooth one contains a slimy polysaccharide capsule. It is far more pathogenic, meaning that it can cause fatality. It is much more virulent. Type 2R, the rough strain, which is a different form of the same bacteria, does not contain a capsule. So as you can see on this image, the smooth strain has an outer coating that can prevent the white blood cells from detecting it. However, the rough strain, on the other hand, doesn't have that, in which case, because it lacks a capsule, it tends to be non-pathogenic. What that means is that if you were to get infected by this particular strain, you may get sick, but you don't necessarily die from it. And we call that an avirulent strain. So what are the key differences between them? Take a look at this. Here's a simple schematic diagram, if you will, that shows what these two bacteria kind of looks like if you were to examine them under the microscope. This is what the smooth strain looks like, and this is what the rough strain looks like. Now, in each one of these, the key point is that the immune system has to find a way to detect the presence of the bacteria so that it can develop the antibodies against it to tag it, congregate it, and then attack it and destroy it. But if there's a capsule outside of it, then it becomes very difficult for you to make the detection properly. There was a famous medical doctor known as Dr. Frederick Griffith in 1928 who actually experimented with both types of bacteria, the type 3S smooth strain and the type 2R Streptococcus pneumoniae. Now, prior to his work, bacteriologists at the time believed that these types are fixed and they are unchangeable from one generation to the next. But his work provided the first evidence that genes are molecules and that things can change. In other words, that these bacteria are not fixed to stay at the form that they're in. And he provided some of the first evidences of that, which no one else had knew about. So how did Griffith's work develop a better understanding of the type R and type S strain bacteria? So this is what he did. He took a bunch of mouse, in this case mice, and he injected them with the living R strain. So these are live bacteria that you injected into the mice, and the result is that the R strain does not cause pneumonia, in which case the animal would survive and no problem. But in another set of mice, what he did was he injected them with the live smooth strain bacteria. All right. In this case, the mice, all of them developed pneumonia and they all died, which helps establish the idea that the smooth strain is virulent, whereas the rough strain is avirulent. So what happens if you were to apply heat to the smooth strain bacteria? So what it does is that the heat would actually destroy the capsule on the smooth strain. So what it does is that it turns a virulent strain, the smooth strain, to become avirulent, meaning that it allows the immune system to detect the smooth strain bacteria and prevented them from multiply at such a significant number. So then what happened? The end result is that when you inject the heated kill smooth strain bacteria into the mice, it won't cause pneumonia and all the mice gets to survive. Now, the most significant part and the surprising part of all this is the next step of the experiment. So far, you know that if we were to inject the live rough strain, the mice would survive. If you inject a heated smooth strain where the bacteria has died and the capsule is removed, the mice also survives. But now what happens if you take the live rough strain bacteria and you combine it with the heat killed smooth strain bacteria? It turns out when that happens, the mouse would develop pneumonia and die. Now, if you base it on purely observation of what you have seen in the mice so far, you know that this should not have happened. As a matter of fact, when Griffith conducted his own experiment at the time, he thought he made a mistake. And he went back to look at his own lab notes and he repeated the experiment several times. But regardless of the number of times he repeated the experiment, the final outcome was the same. In every single group, the mice died. And when he took a look at the bacteria under the microscope, to his surprise, is that he began to find that there were some live strain bacteria that you can see in the mice's body, but he never put the live strain in it. So how did it happen? And that became one of the biggest questions that needed to be answered in order to explain this observation that he knows is real. So this is Griffith's conclusion, that somehow the rough strain acquired the ability to produce a polysaccharide capsule, which is that smooth capsule that I had mentioned to you at the beginning of this lecture. He described this as a transformation, meaning there was a change. And the live rough strain somehow developed an ability to produce the smooth coat that the smooth strain bacteria was able to do on its own. But yet, rough strain should not have been able to. Now there is an interest that has been developed where the scientific community wanted to understand. Now, before I tell you that part of the experiment, I want to show you something really exciting. Last year, towards the middle of June, this particular article was published. And what it shows you in here is that bacteria are using their pili, which are microscopic legs that they have, to stretch out to try to grab genetic material from their surrounding, and then they would incorporate it into their body. Now, what I'm showing you here on this slide are actually just pictures, but there's a real video clip that was uploaded online by clicking on this link, and I hope that you could go and take a look at it. And of course, I will put this video clip on the description portion of my lecture so that you can click onto it and take a look at it for yourself. And this is the first proof that we've had where we can see this in real time. Okay, let's go back to Griffith's experiment. So if you think about the series of major science developments from Gregor, Boveri-Sutton, Morgan, and now Griffith's work, why is this study that he had just completed be worthy of attention within the scientific community? If someone can figure this out, what kind of implications can it have? Remember, up until the time when Griffith was doing experiments, other bacteriologists believe that different types of bacteria remain fixed during their lifetime and that they cannot be changed. So what are the implications if we can prove otherwise? What do you suppose scientists in the field did next? So let's try to imagine for a moment if you are some of these scientists in the field during this time period. The question is, how would you identify the transforming factor in the smooth strain of Griffith's experiment? Okay, so what's causing it to change? Now, Avery, MacLeod, McCarty were the team of 3 scientists that that figure out the transformation factor that led to Griffith's observations. And they actually won the Nobel Prize in 1944 for the discovery that they have made. Let's take a look at how would this work. All right. Now, first of all, you would take the live strain bacteria that you have around you, then you would provide heat to destroy the bacteria, then you would homogenize it. What that means is you would blend it and you would break it apart. Then that becomes what we call the virulent filtrate. So this virulent filtrate will then be comprised or composed of cellular components and molecules inside the filtrate. But can you figure out the fact that you are blending a whole bunch of smooth strain bacteria? What would be inside this Erlenmeyer flask that makes up the filtrate? What you have now is a flask that contains proteins, lipids, RNA, DNA, and carbohydrates. Now, here is the important question for you. What can you do experimentally to find out which one of these components was the transforming factor in Griffith's experiment? Now, there are 5 major categories of molecules here. What can you do to figure out which one of these 5 is responsible for changing the rough strain into become a live smooth strain? There are essentially 2 important questions that you need to be able to answer. Question 1, how would you isolate each one of these components when you cannot physically separate them? Back in the early 1900s, we did not have the technology to physically separate and isolate and put all of these proteins into one test tube, another, the lipids, and another one. You simply cannot do that. So if you can't do that, can you think of a molecule that you learned last semester that can help you achieve this? Is there something you can add to this jar or in separate test tubes such that you can knock out some of these molecules, even though you can't literally physically remove them? That's question 1. Because if you can't isolate each one of them, right, like the way you would try to design a good controlled experiment, then you won't know which one is causing the effect that can help explain Griffith's observations. That's step 1. Step 2, how can you test and show that one of these components was the transforming factor? So in this particular case, after when you isolate them, you need to then be able to verify that this is in agreement with what Griffith had observed to explain what had happened. How are you going to do that? What kind of instruments would you need in order for you to provide the proof? So let me walk you through this one step at a time. So those are the 2 questions that I have re-put on the top of this slide. So it's the same 2 questions, just to keep you in mind. All right. And now let's take a look. One of the things that you can do is to add, you're right, something you learned last term, an enzyme. You can add specific types of enzymes to knock out one of these 5 major components that is found in the smooth strain bacteria, one at a time. And the way you do this is that you take some of the filtrates that you already have from breaking down the smooth strain, you put a little bit of this solution, which in this case was colored in red. It's just an arbitrary color that we used. And you put a little bit of that into the test tube. Then you add the enzyme lipase into it. And when you add the lipase into it, what then happens is that you remove the normal functions of lipid. You destroy it. You prevent it from doing its normal thing, leaving behind 4 other molecules that are still in its active state, supposedly in its normal function. Then you add the live rough strain bacteria into that same test tube containing the 4 remaining components that are still functional. Then you wait and watch to see if the live strain will be transformed into a smooth strain. Remember the instrument I asked you? How can you verify it? Well, how do you think Griffiths was able to, or other scientists were able to name the bacteria as such? They used a microscope. The reason why type 3S and type 2R bacteria are named as, in generic terms, rough versus smooth is because of the texture and contour that it looks like under the microscope. So in this case, if you want to verify if the bacteria that was growing inside this test tube right here, whether it contained either rough or smooth strain, you would put it underneath microscope, and that would show you where the transformation had occurred. Now, the whole idea behind this particular approach is the fact that enzymes are highly specific. Now, a lot of times students would ask, well, how come they don't put maybe 4 different enzymes into one of these test tubes so that you knock out 4 and leaving 1 behind? Isn't that easier and clearer? But there's a problem, right? Think carefully what you know about the characteristics of enzymes. Every enzyme functions at a very specific range of temperature. There's optimal temperature, there's optimal pH level. So if you try to put 4 different types of enzymes into one test tube, you won't be able to get it to function properly, which is why this group of scientists only added 1 enzyme into this test tube right here at a time, knocking out 1 of these organic molecules at a time, leaving the other 4 behind. The idea is this, is that if you knock out lipids out of its normal functioning capability, and if transformations still occur, what that shows is that lipids are not responsible in transforming the rough strain into a smooth strain. Now, that's for knocking out lipids. You use lipase. Can you think about what other enzymes you would use for the remaining 4? So how many test tubes do you think they had to set up in order to answer the first question? Which leads to the observations from the second one. Here is the answer. This is what Avery, MacLeod, and McCarty did. They had set up 5 different experimental groups. In each one of these, they added different enzyme. RNase in the first one, protease in the second one, DNase on the third one, lipase on the fourth, and carboase on the 5th one, right? In each one of these, what you're doing is you're eliminating 1 out of 5 organic molecules that are inside your filtrate, allowing you to see, after when you add the live rough bacteria into here, to see if transformation occur. Now, if you look at this carefully going across each one of these groups, when RNase is added to the test tube, transformation occurred because you know what happened? When you put it under a microscope, you can find smooth and R strains, smooth and rough. You find both. Now remember, live smooth strain was never put into this original test tube. You only added live R strain bacteria, which means this S strain came from transformation, which means knocking out the RNA did not stop transformation from occurring. So in other words, RNA, it's not responsible. Now, if you take a look at the second set of experiment, they added protease, an enzyme that destroyed the function of proteins. And what happened? Smooth strain bacteria and rough strain bacteria can still be found. In other words, transformation occurred, regardless of the presence or absence of this protein that was originally in this filtrate. Again, this is not what you're looking for then. When they added DNase to destroy the DNA inside the smooth strain filtrate, and then they added the live rough strain, they found that no transformation had occurred because all that you can find was the rough strain bacteria. Notice there is no smooth strain. In other words, by destroying the DNA, you cannot make transformation happen. This stays as the rough strain bacteria, just like the way you added in at this step. Now let's look at the remaining ones too. When you added lipase, the result is that transformations still carry on. Again, lipids are not responsible, regardless of whether they function or they don't function. In the last step, they added carboase to destroy the carbohydrates, And again, transformation occurred, regardless whether they're functioning or not. And the fact that in 5 of these experimental groups, only the third one where no transformation occurred, this is how they were able to isolate and verify that out of these 5 contenders, DNA is most likely the hereditary material that actually allow transformation to occur. Isn't that exciting? Now, I'm going to come back to this point a little bit later. But here's the thing. The race continues. The scientific community was not totally convinced that DNA is the hereditary factor. Why? Because the scientific community says that what they did was that they showed that DNA was the transforming factor in bacteria, but not in all other types of organisms. Well, so then what do you think happened next? What would you do if you were the other competing scientists? Knowing that the explanation, yes, they provide a solid proof, but not for everything. What type of experiment would you perform to verify if DNA is in fact the hereditary factor? Well, another team of scientists by the name of Alfred Hershey and Martha Chase, in 1952, they used bacteriophages and bacteria to conduct the experiment. Now, there is one thing that you need to keep in mind as we conduct this part of the talk. As you know, viruses cannot self-replicate. They always need to infect a host. So there must be something that the virus must be injecting or putting into the bacteria cell in order for it to replicate itself. So what would that be? So what Hershey and Chase did was that they tagged the virus, different parts. And again, a lot of the suspicion is that at the time, that protein is probably that there's a good chance that protein is a hereditary factor. Why? Because if you recall the basic structure of an amino acid, there is that letter R, also known as the variable or the side chain. You can have a lot of different molecules that can attach to it, allowing it to have a wide level of capabilities. It can change into many different functional molecules. So there's a strong belief at the time that protein is probably the hereditary factor because it allows an organism to develop all of these different traits. And DNA was a good contender in part because of what Alfred and Chase was able to show in their experiments. So what they did was they need to find out, is it protein that enters into a bacteria cell? Now in this particular diagram right here, the circle that is outlined in gray is representative of the bacteria cell. So what they want to know is it the phage coat, the capsule right here that enters the bacteria cell, or is it the genome, the DNA that is inside the phage coat inside of it that actually enters? Now, what they suspect is that whatever actually enters the bacteria cell, then that's the genetic material that you're really looking for, that you can say it is responsible for the genetic material that can be passed from one generation to the next. So what did they do? Well, Hershey and Chase used 2 different tags. They used a phosphorus tag, P32, to label the DNA. Then they use a different one known as the sulfur 35, and they use that to tag the protein code. Now, in this case, the sulfur 35, it's identified using a red color, just to illustrate the point, and the phosphorus 32 is used to illustrate the tagging of the DNA. In this set of experiments, you take one group of the bacteriophage, you let it infect a bunch of bacteria, and then you let it grow inside a petri dish. Then you put it into a blender, then you break it apart in the test tube. Afterwards, you pour it out and you check to see where it is that you would find the DNA material. Now, if you take a look at the example for the phage on the bottom, the scientists labeled this with a sulfur 35. Let me just clarify one thing, is that both of these molecules, the phosphorus 32 and the sulfur 35, these are actually radioactive isotopes. And the reason why they chose sulfur 35 is because uh, the amino acid code right here contains sulfur. So by tagging it with this sulfur 35, you can use it as a trace marker to see and to track what happens to it after when you allow it to infect the bacteria. And the scientists chose to use phosphorus 32, which is another type of radioactive isotope, is because the DNA material itself, the molecule itself, contains phosphorus. So again, you can use that for tracking purposes. At the end of this experiment, what they found is that all of the new bacteriophages that were found inside the bacteria cells, they were all having radioactive substances irradiating out from them, which means that this particular molecule was passed on from one generation to the next and used to create the new ones. Whereas for the second set of experiments, the one down here that was tagged with sulfur, you did not find any of it in the new viruses themselves, which comes to show that the DNA is the hereditary factor in the phage. Now, at this point in time, the race continues on. Now what scientists want to find out is what is the composition of a DNA. So Erwin Chargaff in 1947 provided more evidence that DNA is in fact the genetic material that we're looking for. But you need to keep in mind, my students, up until this point, no one has seen what DNA really looks like yet. So what all of these scientists are trying to do is, out of the enormous number of choices that they know could potentially be a hereditary factor, they need to first figure out which one is most likely the one before they go digging into what the structure of these molecules may look like. So Erwin Chargaff, what he did was he analyzed the base composition of DNA and then compare it against many different kinds of organisms. And what he discovered is that in all of the different kinds of organisms that he studied, they all contained adenine, thymine, guanine, and cytosine, of which they fall under two different categories. Both adenine and guanine are considered as purines, one type of chemical molecule, and thymine, letter T, and cytosine, designated by letter C, they're all examples of pyrimidines. So this is an example, a snapshot of some of the examples of the data that he has gotten. Humans tend to have 31% of adenines and 31.5% of thymines. Guanine, they have 19.1%, and cytosine have 18.4%. Now, if you look at this chart a little bit more, I want you to pause the video here and use this data to write down your own. What kind of patterns do you see across different species of organisms based on the concentration of adenine, thymine, cytosine, and guanine? So now, let's carry on. What did he observe? Essentially, what he found out is that the DNA composition in species is quite specific. The amount of adenine, thymine, cytosine, and guanine are not the same between species. This diversity was consistent with the hypothesis that DNA is the molecular basis of inheritance. And what he discovered by looking at all of those different numbers is this. If you look at humans, the amount of adenine is almost equal to the amount of thymine. The amount of guanine is equivalent to cytosine. And then if you follow through and look at the fruit fly, Drosophila melanogaster, they are almost equal between A and T and between G and C. Now, if you look at corn, they both have roughly 25% of each, adenine and thymine, approximately 24.5 and 24.6 for guanine to cytosine. In terms of mode, the common mode that we know of, they have about equal amounts of adenine and thymine, about equal amounts of cytosine and guanine. Between E. coli, in E. coli, adenine and thymine is roughly the same between adenine and thymine, roughly the same for cytosine guanine, and the same would apply to a type of bacteria known as Bacillus subtilis. They also have equal proportions of adenine to thymine and cytosine guanine. However, the concentration of each one does vary slightly between different species. So what do the structures of pyrimidines have in common? If you take a look at this particular diagram, you will notice the following. All of these are nucleotides. They all contain a phosphate group, which is indicated by the yellow circle right here. So there's a phosphorus bounded onto 4 oxygen molecules, and then it contains a sugar. Then there is the nitrogen base. All right, so in this case, the nitrogen base is thymine. On the one and down here on the bottom is cytosine. Now, if you take a look at purines, they are also made up of the same 3 parts as a nucleotide. There's a phosphate group, there's a sugar, and then it's bonded to a different base, the adenine, and then the guanine that you see on the bottom. Now that you have taken a look at both both sets of structures, one thing to keep in mind is that the purines are larger molecules in that the adenine and the guanine structures have a 2-ring structure, whereas if you look at thymine and cytosine, they are made up of a single ring structure. So what are nucleic acids? Essentially, they are the components that makes up RNA and DNA. And sometimes other forms of nucleotides can also act as intracellular messengers or coenzymes or for energy storage, as you have learned in the fall semester. Now, this is the extra challenge that I want you to work on. I want you to read the article, The Structure of DNA, Collaboration and Competition. I need you to read the full article. Then I want you to take notes of the discoveries and contributions made by each team of scientists or individual scientists. What did they learn? What did they observe? What was confirmed? And what was eventually published? When were the different discoveries actually made? And whose work was widely publicized? Whose was not? What's important is also the last question, why it was not widely publicized. After when you're done with the reading and you figure out the answers for all of these questions that I have just posed on this particular slide, the last step of this is I want you to create a poster. And what I want you to do is as follows. I want you to create one poster. I would limit this to an 8.5 by 14 sheet. Now, if you want to, because you need more space and you want to make it into 2 sheets of paper where 2 8.5, 11 papers taped together, that's also fine too. What I need you to do is draw a structure of the DNA molecule. Now, there are 2 ways in which you can consider doing your drawing. It's just an example. You could either draw in this type of detail illustration, or you can choose to use a type of schematic diagram that can show the major structures that are within the DNA. Now, you may be wondering, well, why would someone draw something as detailed as this? Because what this detailed diagram allows the students to do is to pinpoint specifically which team of scientists actually discovered which portion of what we know today as the structure of DNA. If you draw it this particular way, yes, you can see that A pairs with T and G pairs with C, but that's pretty much it. You don't get to see the molecular differences between them. So if you want to use it as a way of both challenging yourself and also to learn the details of the molecular structure of the DNA molecule itself, some of the students would choose to draw this. Now, here's the thing. I've also had students who would do a combination of the 2, which means instead of drawing the entire DNA structure in a schematic way, they would draw a portion, a segment of this that shows the detailed illustration right up here on top or somewhere in the middle. I leave it entirely up to you, but the important thing is you need to make sure you include the following that is shown on this slide. I need to have the full name of the team of scientists that actually work together, along with the date, which is the year of the discovery, the research institution and location, And I need you to give me a brief caption of the actual discovery. Make it as concise as you possibly can. And where your grading comes in, that determines whether you will get a high grade or a lower grade for this assignment, is what are the missing links? Which discovery is not described in the article, but should be included in your written work? That, in other words, what were some of the other developments that also contributed towards the eventual understanding and the forming of this DNA model that we know of today? Whose work was it? Where did they work? And when was this discovery made? This is going to be your exit challenge. Okay, so I can elaborate more if you need to. You can feel free to email me if you have some questions. I will set a forum for this last exit challenge in Jupyter Ed so that you can pose each other's questions and I can answer them. And if just in case you all have the similar types of questions, I can pretty much answer once and for all. I can't wait to see what the final product is. And I assure you, you will get a much deeper understanding by the time you are done reading this particular article about what happened in history that ultimately led to Watson and Crick's model for the structure of DNA. Thank you very much for your time. Take care, everyone.